Well, good morning, everybody. Welcome to another crop hour. A crop hour today is going to talk, the information presented is going to be on pollinators in sunflowers. And for those of you who don't know me, my name is Ruth Beck, and I'm an agronomy field specialist. I'm based in Pierre. Just a couple of housekeeping things. I'm just going to cover real quick what you've been hearing, but just in case anyone missed that. As we go through the seminar, if anybody has questions or wants to make comments, you can use the Q&A, which should be on your screen, or the chat feature in Zoom to type those in. But if you choose to, at the end of each of our seminars, we go through we put you all into our discussion and bring you into our room, and then you're able to unmute and ask those questions. So, if you'd like to be part of a discussion at the end of Adam's presentation, we encourage you to do that, and we love to hear those questions. So feel free to unmute and do that if you'd like to. Our presenter today is Adam Varenhorst, and Adam is an extension entomologist at SDSU. He's been there since August of 2015. He grew up on a farm in Iowa and graduated with his BS from Briarcliff University in Sioux City, Iowa. And in 2015, he graduated with his PhD in entomology from Iowa State University. So Adam's been with us about 5 years and is done a great job as a state entomologist. So we're going to welcome Adam.
Thanks, Ruth. And thanks for the introduction. So good morning, everyone. Today I am going to be speaking on sunflower pollinators. I'm going to be highlighting some research projects my lab's done over the last few years, and then just talk about what our findings were. So if we think about pollinators, there's really the two big types. There's managed pollinators, which in South Dakota really is focused on honeybees, although in some cases bumblebees can also be managed. Typically these are more for greenhouses or growing tunnels. So for most of our field crops, when we think of managed bees, we're thinking honeybees. And then we have the non-managed pollinators, which is pretty much everything else. And in South Dakota, that also includes the bumblebees. So anything that we aren't actively trying to manage the populations of, in terms of we have colonies of them that we move around, those would be considered the non-managed. And if we think about sunflowers, in central South Dakota, we have quite a few acres of sunflowers every year. And we think of sunflowers as being this huge source for pollination, but do sunflowers need pollination? If we think about that question, well, wild sunflowers, you will see a lot of pollinators on those. Those do require pollination. However, our commercial sunflowers do not on the same degree, because the breeding for these varieties over the years has really focused on developing self-pollination in our commercial varieties. And so you're thinking, well, how do the two mix then? Well, sunflowers, because of the acreage and the fact that they are a very large flower, represent a major resource for pollinators in South Dakota. So as the picture shows, if you're out in the field, the sunflower field, while it's flowering, you will see bees on the head. And typically they're going to be pretty active. They're not just going to be sitting there. They go in their circles. And research has observed that the bees actually, because the flowers start on the edge and work towards the centers, you can see the centers not filled out here yet. The bees start on the outer edge and work their way in too, as they're working on pollinating that plant or just gathering the resources from it. And so we had some questions, particularly what pollinators are actually visiting sunflowers. Are we seeing more honeybees, or are we seeing more native bees? A lot of previous research suggests that the native bees can really rely on sunflowers. And then there's this field of research that also looked at the fact that when you have honeybees plus native bees, sometimes the native bees encourage the honeybees to be a little bit more productive. It's probably a factor of competition there. And there's also, looking at the literature, there's a little bit of evidence that maybe honeybees and native pollinators could improve yield for confection sunflowers. And so that was a question we had a few years ago. And this study was headed by Jared Persifka from the USDA in Fargo. And he had a postdoc, Rachel Mellinger, working on this study. As you can see, in South Dakota, we were a collaborator. But the whole purpose of this study was looking at different confection lines and seeing whether or not pollinators would improve yield. And so what we did was we had sunflower heads that were covered with netting so that the pollinators couldn't get to the heads, but the netting allowed air to move through it so that we didn't end up with very diseased heads. And then we had heads that were open so the pollinators could freely visit. And those, we just randomly marked some non-covered heads. We harvested the covered and the uncovered heads that we needed and compared the total number of seeds by weight. And if we look at these graphs here, so this is seed mass per flower head. So this is the averages for North Dakota, which are the first bars here, Nebraska, and then South Dakota. The dark bars being closed and the lighter gray being the open ones, the ones the pollinators could visit. We see that in both 2016 and 2017, pollinators significantly improved sunflower yields. And we see that for each state, although we do see that there are some differences by state, and that could just be due to the pollinator composition, whether or not the varieties were well adapted for those areas. But the big takeaway is it looks like for confection sunflowers from this study, there's some pretty clear evidence that there's a benefit. And some of the theories behind that is because, as I showed that picture where the center of the head wasn't fully filled out, there's a belief that these pollinators are actually helping the head fill out 100% or as close to 100% as possible. And so, you know, a yield increase of about 45% could be very substantial. What was interesting in this study, and most of the pollinator data collected from this study is from North Dakota, they were seeing all these native species. They weren't really seeing honeybees. They were seeing these really small native species. And so that got us interested in what we were actually also seeing in South Dakota. And so we conducted an experiment where we collected data for 3 years just in South Dakota. And if you look here, each year our locations in central South Dakota varied a little bit. That was based on where we were having the sunflower variety trial. And so we borrowed some space from that trial and then included our sunflower plots. In 2016, 17, and 18, though, you'll notice we always had a location in Volga. You're thinking, I mentioned Volga yesterday if you were on, why does he keep growing sunflowers in Volga, South Dakota? Because that's not really where we see the production of these. Well, it's a really nice comparison, though, to see if we have specialists where sunflowers are routinely grown in the pollinator community versus where they really aren't. And so that was the reason why we included the Volga location. We wanted to compare what species we are seeing. And so each year we had commercial and also oilseed varieties. So the confection varieties that were used in that previous study I showed the graph from, most of those weren't commercial varieties. And so we wanted to see, well, if a little bit more breeding efforts were put into the lines, what do you see? Do we still see some benefit, or maybe we don't? And we are also curious, is it just confection sunflowers that see this benefit, or is there also a benefit maybe for oilseed varieties by really promoting that pollinator activity and whether or not we are going to see it? So for the oilseed, I don't have company names here, but here are the varieties we use. We use P63 and Hornet. Those were ones that we were able to obtain. And then for the confection, we use 5009. And so these were more or less randomly chose based on if we could get our hands on them. And so we didn't really have any favorites. These were just ones that we sought out and we were able to get. So at each location that we conducted this study, we had 10 blocks. within those blocks, we had those 3 sunflower varieties. So we ended up with 30 plots per location. And the reason we did so many blocks was because without a lot of treatments, the varieties, we felt like we had a little bit more time. So we wanted to really make it so that we could get really good data each year. And we planted the sunflowers at 20,000 seeds per acre. And then each year, we made sure those sunflowers were planted about mid-June. We started scouting before the heads, before the plants reached flowering. So when we started seeing buds, we really started scouting, and we got everything prepared a little bit before that to do our sampling. And then the heads were harvested sometime normally in mid to late October. Now in each plot, we used a very similar method as that previous study where we bagged and then also marked heads. The marked heads, we used just marking tape. We wrapped it around the stem a little bit below the flowers so we wouldn't have any impact on the pollinators. That just made it so we had randomly selected heads for when we were going to harvest. And we've had those before the heads developed, so we weren't accidentally or arbitrarily choosing really big heads. We wanted to make sure that the heads weren't developed when we marked them so that we weren't being biased in looking for the really huge uncovered heads. And so this is what those nets look like. So we actually used, it's referred to as no-seam mesh. So it keeps insects out, does allow air to move through. And so these were custom sewn nets, and then they were tied down at the bottom with zip ties so that nothing could go up into them. We had to make, you see there's quite a bit of baggage on this head right here. The reason we did that is during that first study, we ran into issues, especially at the Volga location during years where we got quite a bit of precipitation. We were getting confection heads that were, they were huge. They were maybe 15 to 16 inches across, sometimes bigger, and they are actually tearing apart the bags in that first study. So we actually lost some of our plots that year because the heads just grew so fast and so large that they tore the bags apart. So we made sure for our study we had larger bags so that the heads had a little bit more room to grow in them. And so to collect the pollinators, I will say this again, is we can't really use sweep nuts and sunflowers. You can see the architecture here. If you try to hit the head here with a sweep net, they'll probably either break it off or it'll come swinging back at you and hit you in the face. And also the leaves aren't going to be able to tolerate a sweep nut sampling inside. Even though we'd be aiming for the heads, we'd cause a lot of damage. And so instead we use what are referred to as bee bowls. And so here's what we use. We use the fence post and then PVC pipes that were cut down, and then they have a coupler up on top. And that coupler is big enough that these little cups can fit in there, and these cups are painted with fluorescently dyed paint. And so the yellow and blue with the fluorescent dye in there mimic flowers. So it's going to be very similar to what the bees are actually looking for on a sunflower or other flower when they're looking for resources. And so we put those right at the sunflower head height. If you notice, there's some stuff floating. The picture's a little bit blurry. I apologize, but we actually put soapy water in there. And so it's not the most, not the best practice maybe for preserving bee populations, but it's how we can sample them. And so the whole idea is the bees are here at the flowers. They see our painted cups and they think, oh, that's another flower. They fly in to check things out. They hit the soapy water, and the soap covers them and also prevents them from then flying back out. And so we capture them, we go through and collect them later. So when we put those traps out, we'd then go, once the heads, as soon as the sunflowers started flowering, we'd put our little cups with the soapy water out, and we'd leave them in the field for 24 hours. And then we'd go back and collect them the next day. And then we did that on a weekly basis. Now we could have sampled more, but one of the issues we had was most of our sites were in central South Dakota. So 2 days a week, driving a round trip of 6 to 7 hours really adds up on the finances. And also we didn't want to oversample those pollinators because we didn't know how robust those populations were. We didn't want to decimate them. Once we collected the bees, we put them in to those little cups. We actually had lids for those. We put them into a cooler, or a refrigerator on campus. And then within a week, they had to be washed and sorted. And then we didn't have to identify them right away. We actually froze them once they were all prepped until we had time to identify them. And so I said we had to wash the bees. We call it the bee salon. We're borrowing that term from the lab I was in at Iowa State. They did this type of practice too, but the soapy water mats the hairs of the bees down. And the big issue with that is when you're trying to identify a pollinator, you actually have to look at the hairs a lot of times. And so it's very important that they are fluffed up like they would be when they're alive and be able to fly around. And so we used a sieve here. We'd spray in, this is just water. We'd wash that soap off. As you can see here, there's the soap being washed off. And then we'd blow dry the bees, because if you want to fluff up a hair on an insect, the best thing to do is to use a blow dryer. Now, this had a little mesh covering over the jar, so we implemented little mesh, they're metal mesh, but so that it could stand a little bit of heat, but not too much. And then we used the blow dryer typically just on low with no heat, because if you do use the heat, since it's an enclosed area here, you're going to cook your bees. And so that was something we also didn't want to run into. But we'd sit there, blow dry the bees so that the hair fluffed up. And then we'd lay them out so that they could finish drying, as shown here. And we always kept the labels. Once they were fully dry, we'd put them into these little vials here, into marked boxes, and store them in the freezer until we went back to identify them. And as long as they were dried 100%, the hairs stayed nice in the freezer. I thought it was just kind of interesting. We talk about collecting and working with bees, but sometimes maybe you don't know all the other things we have to do to try to get those to the level where we need them to identify them. It's a long practice, but it does make it a lot easier for us to do our end job of identification. So in 2016, we collected a total of 754 pollinators from the Volga location and 524 from the Oneida location. And if we look here, Hymenoptera is the order for bees and wasps. Diptera are flies. And so we saw that the majority of the insects we collected were bees. We had quite a few flies at the Volga location. If you've ever heard me talk about natural enemies, there are these insects called hoverflies, and the larvae of those actually feed on aphids. Our sunflower plots at Volga were next to our soybean plots, where we had soybean aphids, and we think that we had influenced our hoverfly or populations a little bit, because we had the prey next door to the sunflower, and then the adult. So those flies are pollinators, so they were really enjoying the sunflower plots next door. But it looked like most of what we collected were bees. And it looks like, there we go. Just didn't load right away. So from our 2016 species, so these were out of the totals that we collected. Here are all the different species. So we had quite a few. If we start looking, this is what I wanted to point out. So we had a lot of flies at Volga because of what was next door to them. But some of the Andrena species contain some sunflower specialists. They're just really hard to identify though. We had 117 individuals of those. And Melisodes trinotis, and I apologize, they're all species names. I didn't do this just to be really technical. Most of these bees don't have common names. Apis mellifera are honeybees. We just kept it consistent. Most of these bees don't have common names. So we left the species in there just to make it easier to talk about. Melisodes trinotis is another sunflower specialist. And if we look, we actually collected more of those at the Oneida location. And overall, we had quite a few species that we are observing at both locations. One of the things I like to point out is we had honeybee hives relatively close to these plots at both Volga and Oneida. And we didn't see a lot of honeybees. A lot of research has shown that honeybees are too smart to go into bee bowls. They know what our tricks are. They know that those aren't really flowers, and they just avoid them. So researchers have put bee bowls right next to really good resources where it's just covered in honeybees, and the honeybees just avoid the bee bowls. So a few will go in to check it out, but the majority say nope and will have nothing to do with it. So our most abundant pollinator in 2016 in South Dakota was Melisodes trinotis. It made 64% of the total population. One of the interesting things, I didn't have a picture of the honeybee, I should have had it side by side, is these look pretty similar to honeybees just because of the brown bodies and the markings. If you ever are trying to determine whether you have a honeybee or another species, look at the eyes. If you're having a hard time, because honeybee eyes will have little hairs on them, other species won't. So that's one of the characteristics that can help identify honeybees from other species if you're kind of unsure of what you're looking at. In 2017, again, excuse me, we had the majority of our samples being made up of bees and wasps. And this time we had our location in Highmore. We had a lot more pollinators collected at Highmore than Volga. We didn't have soybean aphids in 2017 at our Volga location. I think that really shows the difference that we are seeing in 2016. I'm not sure, there we go. I'm not sure what's happening with my slides. I apologize. It's there, but it doesn't want to show up right away. For 2017, again, we saw that we had more of the specialist and the Adrena species at the central location in Highmore. We also had more of the Melisodes trinotis. This year we did capture more honeybees, but still a very low percentage of the total population. We also had some other species, so lazy wasp, these are another small species and can be specialists and can feed on sunflowers, so they can sometimes seek sunflowers out. But our total and most abundant pollinator again in 2017 was Melisodes trinotis. It made up almost 29% of our total population. For 2018, we collected, again, a lot of flies at the Volga location. Guess what? We had soybean aphids again. But most of our bees actually came from our Gettysburg and Pierre locations, if we combine those. In our 2018 results, we had another location. We just were trying to make our sampling a little bit more robust. But if we get down to our species again, so we had Melisodes trinotis, and it made up quite a few individuals. We had some other Melisodes species show up that we couldn't identify, and bees can be very difficult to identify, especially some of these smaller ones. There just aren't a lot of characteristics that can be used to tell them apart. You need specialists. The problem with that is the specialists charge a lot of money in some cases to identify each individual, and also they are very busy because there aren't a lot of specialists in the country. So our take home though from our 2018 results was, again, in 3 years, our most abundant pollinator was Melisodes trinotis in 2018, almost made up 25% of our total collected individuals. One of the big questions we get asked when we're doing a survey like this, where we're trying to determine what species are present, is did we actually sample enough to be able to confidently say we saw everything that should have been there? And so one of the things we can do to answer that is to construct what are called species accumulation curves. And so what this is we take our total individuals or samples that we conducted, and we look at the total number of species we collected. So this first mark here, the dark line, actually represents the accumulation of the species we saw. When that dark line stops, that's the max number of species we saw. And then we see this dotted line. That's an extrapolation based on what we saw. This software we use constructs this next line by analyzing our data and calculating what is possible. And it tells us how many additional individuals we would have needed to collect and what maybe our max number of species was that we could have observed. And so we look for when this line kind of stays flat, so it stabilizes. These represent our confidence intervals, and so we'd like those to be kind of flat. The farther they get away from our line, the more chance for error based on the calculations there is. And so from 2016, our top two graphs here represent Volga and the bottom two represent Oneida. The big thing to point out is when it's blue, has the blue lines, that's the number of samples. And when it's red, that's the number of individuals. And so we observed 13 species at our Volga location in 2016. And we had about 125 samples and somewhere between 600 and 700 individuals. What this is showing us is that we could maybe have gotten to about 15 species, but to do so, we would have had to just about triple or a little bit over, maybe triple our sampling somewhere in there to get there. And then if we look at the individuals, we would have had to collect about 1,600 individuals. So we could have gotten a couple more species, but we would have had to really increase our sampling so that we had more samples and we collected a lot more individuals. One of the nice things about these is it gives you an idea about the economics. So if we had to increase our sampling that much, it wouldn't be feasible to only feasible when we're only looking at maybe getting 1 to 2 more species. And so that's what these are also really useful for. So we can make the argument we probably sampled enough at the Volga location because we weren't going to maybe see as much. If we go to our Oneida location down here, we observed 10 species, and it looks like we maybe could have gotten 16 if we would have sampled about 225 samples or 3,000 individuals. One of the issues here is if you look at these bars, the air bars get really large, so maybe there could be some more. So at the Volga location, we could have maybe sampled a little bit more. However, as I mentioned, those are about the Oneida location, the Oneida location was about 3.5 hours away. And so that made it a little bit more difficult to get a lot of extra sampling in. Our 2017 results, again, the Volga location's up on top and Highmore's down here on the bottom. In 2017, we saw 11 species at the Volga location. We could have maybe gotten up to somewhere around 14. So again, at the Volga location, we probably had a pretty good sample size and collected enough individuals to see what species were present. However, if we look at our Highmore location, we collected about 25 species. So we collected a lot more species in central South Dakota. And we could have maybe gotten up to about 35, 36 species had we increased our sampling to about 525 samples and just about 2,000 individuals. So maybe in this case that would have been feasible in terms of the number of extra species, but if we look, there's a lot of error associated with that, so we may not have actually seen that many extra species. In 2018, the top is now Gettysburg. This is the Volga. If we look here, the Gettysburg location, we had 22 species. We could have maybe gotten to 30, but it would have taken a lot of extra samples and individuals. And then for the Volga, we had 20 species. We could have maybe gotten to about 50. However, I don't trust that because if you look here, the standard error, the confidence intervals are huge. And so probably aren't going to see that many. And so this may just be an artifact. I'm guessing we actually sampled enough in that year. And then at our peer location here, we had 20 species and we could have maybe gotten to 30, but we would have needed to increase our sampling and the confidence intervals are pretty wide. So there's a chance that we won't actually see that many extra. So all those sampling species accumulation curves I showed indicate that we probably did enough sampling. We could have maybe increased a little bit more in 2017. However, the big thing with sampling is you have to get as many samples as you can to really see how many species, but you also have to stay within your budget. And so sometimes to get those extra species, you'd have to exceed the budget, and that's not going to work out. Melisodes trinotis, which is a native pollinator, it can specialize on sunflower, was the most abundant pollinator every year. We just didn't see a lot of honeybees. We did see some in the field, however, we just didn't collect a lot. And so our central locations had much more diverse populations of pollinators than our Volga location, which was very interesting. That showed us that we actually do see a lot more specialization probably where we grow sunflower. It's not a huge surprise, but it is really interesting the fact that we do have those pollinators present where we are growing the sunflowers. So I mentioned yield, and I'm going to show that to you. So for 2017, our Volga location, these are sunflower yield per 6 heads. So you look, it's very small values because it's in pounds. But here's our confection variety in the light gray. And we saw that there was a significant improvement to having pollinators visiting that variety compared to having them closed off from it. If we look at our hornet open and closed, we saw a little bit of a benefit. And for our other oilseed variety, again, we saw a little bit of a benefit. For our 2017 Highmore location, we really didn't see any differences. It actually looked like in some cases the closed varieties yielded a little bit better, but they weren't significant. For our 2018 Volga, we again saw that the confections, when they were open, yielded a little bit better. We actually saw that the closed hornet, so one of the oil seeds, yielded better than the open, but our other oil seed yielded better when the pollinators were visiting. We should have had data from 2018 in the Pierre location. However, there's a major hailstorm that you could actually see the scar from satellite, so where the hail hit. And our plots happen to be right there in an area where it got hit really hard, so we just didn't have a lot left to harvest after that happened. So conclusions. For pollinator visitation, we had improved sunflower yields with few exceptions. We aren't sure what happened to those cases, but we saw improved yields in Volga, and maybe there are environmental factors influencing that, including maybe the extra moisture helped. And one of the issues when we put coverings overhead always is if that influences, and hopefully we didn't by making the netting as big as possible, but something to always consider. So that's all I have for you today. And I'd like to thank the funding sources that helped out with these projects, National Sunflower Association and South Dakota Oilseeds Council. We really appreciate the funding and continued support. And then if you have any questions, I'd be happy to answer them today, or if you'd like to call me, or if you have future issues come up that you have questions about in your sunflower or other crops, please feel free to give me a call at my office number or send me an email. If you ever have an insect identification that you need, you can always send me a picture of it to my email, and I'm normally pretty good at identifying those. It's great that our cell phone technology has improved so much over the years, because the cameras on your cell phone now are a lot of times better than our old digital cameras that we used to have. So technology is a great thing for insect identification because it makes it really easy for us to see what you're seeing. So Ruth, I'll turn things back over to you, and if there's any questions, I'll be happy to answer them.
Yep, I'm gonna just pull up the QR code for people. So if you want to get your CCA app, if you're a CCA on open, and then you can scan the code. That was really interesting, Adam. I didn't know if you could just, really quickly. I don't recall. I don't recall hearing anywhere in the last 2 days, information for people about, how we can avoid, maybe reduce our impact on pollinators when we do spray insecticides on sunflowers. You might have said it, I might have missed it, I apologize, but I always think it's good to hear that. So I don't
Yeah,
know.
I actually didn't have it in there, Ruth, and one of the reasons is that we don't have what I consider a great solution to that issue. If we think about it, though, most of the insecticides we're using will have negative impacts on the pollinators. And so if we try to avoid times when the pollinators are most active, so typically we say try to spray later in the afternoon, evenings. In some cases, though, it's very species dependent. And so some species can be more active at those times than maybe at noon. But it's just really tough. And so for pest management and sunflowers, we know we have, like I mentioned yesterday, red sunflower seed weevils, banded sunflower moth, sunflower moth. Some of these pests that are essentially just a big issue right as soon as we get the flowering head, and that's the time to spray. That's also when pollinators are starting to really seek out the sunflowers. So it's a little bit tough to really have a great answer in terms of that. But the best things to do is if you're out scouting and you're maybe below threshold and there's a lot of pollinators active and you haven't hit the threshold yet, wait until you do hit the threshold. That's really important. As soon as you hit the threshold, though, try, and I know most of our farmers aren't probably applying the pesticides, the insecticides themselves. They're hiring it. And so they're at the mercy of when those individuals can also spray. So, it's something we hope to address better in the future. But now that we know that there is some benefit to the pollinators, we hope that helps us in getting a little bit more momentum going forward to seeing if we can't find some alternatives. Or the timings, like I mentioned, are just tough because pollinators aren't all active right at the same time every day. There's some evidence that maybe spraying at noon would be good because during the heat of the day, pollinators aren't sometimes as active. However, there are going to be some, and then we also have most of our products have pretty long residual periods. So that's another factor. But I guess the best thing I can recommend is try to time it so that it's when maybe the pollinators aren't as active. So most of the time later in the day. One of the things we do, I think that's great in South Dakota is that we have our no-till practices because a lot of our native pollinators are actually ground-dwelling bees. And so when we had our plots, I didn't put the pictures I should have, we actually could see the holes in the field where these ground nesting bees were making their nests out in the sunflower fields. And so it's really interesting, that community that they have out there, because they're making their homes right in the field where the sunflowers are, so it's easier to go back and forth and get the resources. And so with no-till systems, we really encourage those and preserve their populations more than if we had a lot of tillage, because that would disrupt those nests and destroy them.
Matt, I don't know if you want to pull up the survey for people. Thanks. Yeah, that's interesting. I didn't know that about the ground dwelling bees. I know Amanda Bachmann is also on the call today, and she's an entomologist in our office here who deals a lot with people who are concerned about insects in their yards. She's our urban entomologist, and I know she answers a lot of questions about ground dwelling bees in the central South Dakota area. So people who don't care for them. So it's good to know that, they're good. And I don't know if she would want on mic and tell us anything, but
Yeah, I'm
there
sorry to
she
interrupt
is.
you, Amanda. I was just going to mention, Phil reminded me, he sent me a message. He's watching today. Phil is my technician and also the South Dakota IPM coordinator. And he reminded me that before we spray for our stuff, we typically check South Dakota Bee Check. And Amanda, I'd let you give some more information on that, but there are resources to check where honeybee hives are, and so that you are responsible for contacting the beekeepers so that they can keep their bees from going out during your sprays. There just isn't as much we can do for the native pollinators, but I'll turn things over to Amanda to talk about that a little bit more.
Yeah, so for applicators, especially keeping an eye out for the honeybee hives, South Dakota does participate in Driftwatch. It also is Field Check or Field Watch, like all sorts of like the same 3 different names for the same thing. But the South Dakota Department of Ag is encouraging folks, both applicators and also producers. So if you're somebody who produces, say, an organic crop or some other specialty crop that would be sensitive to pesticide application, not just insecticides, but maybe herbicide as well, to sign up and participate in that program. And yeah, one of the cool things with the native bees too is that they tend to be active sort of at more discrete time periods. When we think about things like honeybees and bumblebees, they tend to be active throughout the entire season foraging, whereas some of our native bees are maybe only active for a month or so before those adults are sort of done provisioning their nests and dead, and then their progeny are hanging out in the soil developing. But yeah, I do get a lot of questions from folks in town. They see the holes, they see the bees, and they freak out. And a lot of these bees, since they are solitary, they don't, they're not, and even, honeybees and bumblebees aren't what I would consider to be aggressive. A lot of people think about wasps, and that's where they, get sort of their fear of anything that's orange and black. But these native bees are, fairly docile. I've had, a lot of the sweat bees and stuff land on me when, we've been out sampling in the sunflower fields, and, their first instinct is not to sting. So, they're really a chill species, and we get a lot of calls because of just sort of their number when they emerge sort of all at once. But, they also provide a good service by doing a little bit of natural tillage as far as allowing, water to then infiltrate into the soil farther. So yeah, any questions about bees in the central part of the state, I'm also happy to help out and, get samples to Adam and the rest of the entomology crew.
Yeah, so thanks, Amanda. And I just want to encourage everybody to, if you've got a question, please type it into the chat or the question and answer feature. I think we're now going to go into our discussion. So Matt, who is our tech guy and does a great job, so thanks, Matt, for all your help. He's going to bring everybody into our discussion, and then I encourage anybody who has a question too, if you don't have to type it in, you don't have to take the time, just unmute. I'm going to be watching for mics that unmute, and we'll feel free to speak up and ask those questions. And Adam and Amanda and Phil's on, and Paul Johnson. So any questions that you might have for any of those people, this is a great time to direct them and get them answered. So next week the presentations are going to be on understanding SDSU agronomy research. So if you're interested in that, if you've ever wondered, how the research is performed, or, the sort of protocols that we follow when we do research, join us for some of those sessions next week, same time, Tuesday through Friday, 10 to 11. So. And Amanda just put, the Driftwatch link into the chat box. So if you guys have any interest in, in Central South Dakota, we do have quite a few. I know of at least 3 organic farms right in this Pierre area. So they are popping up, and we have to all be really careful and aware. Whenever I do my private applicator training, I definitely share that information. And caution people about spraying and making sure they know what's around the surrounding areas when they spray. So. Adam, I have to say, since I don't see a question or a chat, after hearing you, I always wonder how we even have bees left sometimes, because, we spray right in the middle. We do our insecticide treatment right in the middle of flowering, but I definitely, hear bees are buzzing almost every time I walk into a flowering sunflower field, before spray, after spray. So they seem to come back and get that pollination activity done. So it's interesting.
Yeah, I don't I don't have any numbers, and maybe that's something we should look at in the future, is to see a pre and post spray, if we see a lot of mortality in terms of bee population. But and Phil maybe could answer this. He's walked through the fields a lot more, uh, really close to the spray timings for some of our research. But maybe there's some behavioral things that the native pollinators do to avoid it. And I know I've visited with some company representatives that, there are products being researched that are maybe a deterrence to bees that could be included in some of our products so that it keeps the bees away from the sunflower or other crops for a little bit after they're applied. But I don't know, Amanda might have more information if any of those are actually currently in any formulations, but I don't know if there are any.
Yeah, I'm not sure if any kind of repellent is in any of our formulations. That would be definitely something to look into. But also thinking about the behavior of the bees. I know from spending a lot of time like with squash, just seeing the different species are active at different parts of the day. And so like right early morning at dawn was when we would see like bumblebees, and then like honeybees would be later in the afternoon. So sometimes I know we kind of have to balance the spray with when the pests are also active so that we can hit the pests. But if you can avoid some of those peak bee activity hours, you can also avoid some of the off-target effects that way as well.
Okay, so we do have a question for you guys, one of you. The question is, do hornets and wasps play an important role as pollinators?
So it definitely depends on sort of the site. I see hornets and wasps in my yard a lot more than I saw them like out in sunflower fields when I was helping out with some of that data collection. And in my yard, they will move from flower to flower because they do use nectar as a food source for them, the adults. But what they're doing a lot of times in sort of a backyard environment is the females are hunting for a food source, like a caterpillar or some other protein source to take back to their larva. So anything that goes from flower to flower is going to be moving pollen around and is a pollinator. Obviously, bees are what we focus on a lot because they're fuzzy, and so that helps them pick up more of the pollen grains. If you've taken, closer looks at hornets and wasps, you'll see that they are not as fuzzy, and so they don't sort of get as much of the pollen on them sort of by accident. But we do consider them pollinators, and they do provide important ecosystem services in the form of being predators as well. So I'm very much like pro wasp. Not to mention like the parasitoids and like the whole like yeah, there's a whole bunch of cool stuff that wasps do.
But that's an interesting question, because when I think back on when I'm in a flowering field of sunflowers, I don't recall a lot of hornets or wasps. I really see a lot of honey of, well, I don't know that, they're all bees. They're bumblebees or honeybees, or is what I primarily see there. So yeah.
Yeah, and if you think about the wasps that we've got like in backyard, like paper wasps are, building their nests sort of like, like in a tree or in eaves or in sort of like elevated structure. And if you think about like if you're in the middle of a field, like away from any, tree, shelter belts or whatever, you're not going to have those nests close by. So you probably wouldn't, I'm not surprised that we don't see a ton of wasps in the middle of a sunflower field.
And I didn't mention it during my talk, but some of our native species, the Melisodes trinotis, are the exception, I guess. But a lot of our native species of bees, Amanda mentioned sweat bees, and some of them are what would be grouped into that. They're actually really small in comparison to a bumblebee or a honeybee or the Melisodes trinotis. And sometimes if we are practicing that, looking for the big pollinators, we might actually miss because they move quickly. They're not really noticeable on the plants, but they're there. They're not so small you can't see them, but our focus a lot of times is on the big buzzing ones, where the smaller ones don't make as much noise. And a lot of times you'll see multiple species of pollinators on a single head out in a sunflower field too, is they kind of cooperate. They stay in their own zones. They don't like to be by each other. If one runs into the other, they both take off, we've seen, but they like to participate in the pollination services. And, you know, if you think about the one head you look at, think about the whole field, there are just so many pollinators out there.
I have to agree. If anybody's interested in that, the place to be is a sunflower field in flower, because it's really just alive. So yeah. And I've never been stung or anything, and I've been through hundreds of fields, so it's all good. But yeah.
Yeah, you take more damage from the actual sunflower leaves than you do from any of the insects.
Yeah, I could relate when Adam said that they fly back in your face, those heads.
Yeah.
Yep.
So I don't know if anybody, you can just encourage you one more time. If you have questions, now's a great time to catch up with Adam. I know he gets busy during the season, and Amanda as well. So, but again, looks like Paul's on, Paul Johnson. So, and he, you know, if any weeds questions too, if he's listening, he could sure address those as well. So, thank everybody, and thanks to Adam for that, you know, great presentation and all the work you do, and thank you to Amanda for jumping in and answering some questions for us. Hopefully everybody can join us again next week on Understanding Ag Research, and share some good presentations and discussion there. I don't see any other questions, so I guess unless somebody wants to take that, I guess we can check out and end a little early today. But thanks again to everybody, and thanks for joining us. Yeah, this was a good week for us, and I'm looking forward to seeing some sunflowers again this year in this area. So
Thanks, Ruth.
Yep. Thanks, Ruth.